Good morning and welcome everyone. What is HEAL? HEAL is a new initiative at Cellectis to develop new therapeutics based on gene editing for hematopoietic stem cells, HSCs. We heal altered genome for unmet medical needs. So far, you knew Cellectis through the prism of our awesome UCAR-T platform for cancers. But Cellectis is much more than just an allogeneic CAR-T company. We're a bona fide gene editing company that has an unlimited reach to a series of diseases. HEAL is one of these new initiatives that unleashes the power of our end-to-end platform. So sit down, buckle up, and watch.
Hello, my name is Patrick Hong. I'm a team leader in Innovation Department at Cellectis. Today, I'll be talking about the process of our genome surgery in hematopoietic stem cell, also known as HSC. Allogeneic HSC transplant is a curative treatment for many human diseases such as hematological, autoimmune, viral, and malignant diseases. For this reason, number of HSC transplant has been steadily increasing each year. More than 43,000 HSC transplant has been performed between 2013 to 2017 in US alone. However, among these, only 43% were HLA matched donor. This creates a potential problem for a patient considering allogeneic transplant because the mortality risk of HSC mismatched donor is increased by more than 2.2 times. However, gene editing offers a unique opportunity to address these challenges with availability of autologous transplant. A single HSC potentially could produce millions of cells in our body every day because HSC from bone marrow can differentiate into many different cell types. If we gene edit this long-term HSC, or as we call it, primitive HSC, we can gene edit all these cell types in the downstream and have these cell types produce therapeutic protein of our interest. Therefore, these primitive HSCs are the target to provide lifetime cure. Just to give you a flavor of HSC gene therapy potential, here are some examples of diseases that can be treated by each cell types derived from HSC. Few examples are sickle cell disease in RBC, MPS I and Gaucher's disease in monocytes, and HIV in T cells. In order to optimally gene edit HSC, Cellectis has developed an optimal process from collection and expansion of patient HSC to perform genome surgery and cryopreservation. There are some features that are vital during HSC genome surgery in order to deliver best gene engineered product to patient. Such key aspects are high modification efficiency, high cell viability, and maintenance of the differentiation potency, increase in primitive HSC, and clinical scale manufacturing. Our process can reach high modification efficiency. For example, we can reach up to 96% gene inactivation rate, up to 78% gene insertion, and up to 75% gene correction rate on different loci. Next, our process allows to have high cell viability and maintenance of the differentiation potency compared to untreated control. What about primitive HSC? We know primitive HSC is an essential target for lifetime cure. We have developed our process to increase these primitive HSC in gene-edited population. As you can see on the left, the markers that identify primitive HSC are increased in Cellectis process. Graph on the right shows more than 4-fold increase in primitive HSC during our process. At the end, our process results more primitive HSC without impacting gene editing efficiency. Finally, we have developed our process into clinical scale manufacturing. Using Cellectis technology, we can perform larger scale gene editing on clinical amount of HSC per genome surgery. Furthermore, these gene edited HSC shows comparable engraftment efficiency in vivo, similar to the untreated control on the right. Once we perform gene surgery, these HSC can be pushed into different lineage in vitro. Here we show that our gene surgery can maintain 60 to 80% gene insertion rate after gene-edited HSC was cultured and differentiated into myeloid, erythroid, and other lineages. In summary, we have developed high level of genome surgery platform. where we can achieve 75 to 96% modification efficiency in multiple loci, and more than 80% cell viability and still maintain differentiation potential. We can also achieve more than 4-fold increase in primitive HSC. We have established clinical scale manufacturing without affecting our key parameters. Cellectis process that we show today opens a new door in development of gene therapy treatment for genetic disease in HSC. Thank you for your attention. Merci.
Hi, my name is Julien Valton. I'm the Vice President of Gene Therapy at Cellectis, and today it's my privilege to present the Thalglobin 01, the first gene therapy product developed by Cellectis to cure sickle cell anemia. Sickle cell anemia represents about 300,000 annual births, 165,000 patients in US and Europe, and more than 5 million patients in India and South Africa. More specifically in US, patients affected by sickle cell anemia represent about 100,000 people, which is 3 times more prevalent than other rare diseases, including Huntington disease, cystic fibrosis, hemophilia, and muscular dystrophy. Life expectancy of patients is 30 years shorter than healthy individuals. The genetic origin of this disease is really well known. It affects the gene named HBB that in normal condition is translated into 2 beta subunits of hemoglobin that associate with 2 alpha subunits to produce the functional hemoglobin. Hemoglobin is expressed in red blood cells and used to vectorize oxygen throughout the body. In the context of sickle cell anemia, a single point mutation in the HBB gene causes the substitution of a glutamate to a valine that eventually leads to the aggregation of hemoglobin and the formation of sickle hemoglobin. These hemoglobin aggregates alter the shape of red blood cells from a well-known donut-like shape into a sickle shape. These sickle red blood cells are harder, stickier, and no longer flow smoothly through the blood vessels. They pile up, sometimes blocking completely the vessels. This phenomenon keeps oxygen from reaching a variety of cells, causing a wide range of symptoms experienced by sickle cell anemia patients. Symptoms are ranging from strokes, skin ulcers, acute chest syndrome, anemia, and debilitating pain events. Today, the majority of FDA-approved treatment address the consequences rather than the cause of the sickle cell anemia. The most widely used treatment is a chronic blood transfusion. Beside that, injection of hydroxyurea, antioxidant, anti-inflammatory, iron chelators, painkillers are also used to alleviate the symptoms of the disease. And today, the only curative treatment is hematopoietic stem cell transplant. It consists in transferring hematopoietic stem cells from a healthy donor to an HLA-matched patient. This approach is really efficient. Clinical results show 91% of overall survival rates and 73% of event-free survival for sickle cell anemia patients undergoing hematopoietic stem cell transplant. While highly efficient, this approach is limited by the availability of HLA-matched donors. In the context of sickle cell anemia, only 20% of patients are eligible for hematopoietic stem cell transplant, leaving the remaining 80% without any curative option. So there is a need to develop new curative approaches to treat the majority of sickle cell anemia patients. We believe that autologous gene therapy represents a relevant option to bypass the hurdle of hematopoietic stem cell transplant. This approach consists in mobilizing and extracting hematopoietic stem cells from patients, correcting the HBB point mutation ex vivo using our TALEN gene editing platform, and reinfusing them into the patient. Ex vivo gene correction would consist in designing a TALEN specific for the sickle cell anemia mutation, cut open the faulty gene, and correct it using a DNA repair template corresponding to the wild-type version of the HBB gene. And this is in a perfect world, but in the real world, things are getting a little bit more complicated because When a gene is cut open by the gene editing tools such as CRISPR-Cas9, zinc finger, meganuclease, or TALEN, this gene is repaired by two different pathways. Number one, the non-homologous end-joining pathway, which leads to the inactivation of the genes, or beta-0. And number two, the homologous directed repair pathway that, in the presence of a DNA repair template, leads to the actual correction of the HBB gene. In the context of sickle cell anemia gene therapy, we want to promote the gene correction and prevent as much as we can HBB inactivation, because such inactivation will potentially generate a collateral effect leading to beta thalassemia. Being aware of that potential collateral effect, we worked really hard to improve the gene editing process to tip this balance towards gene correction. Using our optimized TALEN gene editing platform along with AV6 particles to vectorize DNA repair templates, we established a proof of concept that HSC sourced from sickle cell anemia patient could be corrected. The result obtained on hematopoietic stem cells sourced from 5 different sickle cell anemia patients were astonishing, as we obtained about 60% of gene correction and only 20% of collateral effect. Compared to competitors, our process leads to the highest degree of gene correction and the lowest level of collateral effect, which represents a high therapeutic potential compared to competitors. Because safety is of utmost importance in the field of hematopoietic stem cell gene therapy, we leveraged our knowledge in TALEN design, production, and screening capacity to select the most active and specific TALEN molecule to correct HBB locus. Our unbiased genome-wide analysis of TALEN activity enabled us to show that the thalglobin 01 TALEN was highly specific for the mutated form of HBB, with only one specific offsite identified above the limit of detection. This offsite, located in the HBD locus, was expected and predicted bioinformatic algorithm due to its high level of sequence identity compared to HBB. Quantitative analysis of HBB and HBD gene processing showed that HBD was processed at a very low frequency compared to the on-site HBB. After demonstrating that our gene editing treatment could efficiently correct in HSCs the genetic mutation associated with sickle cell anemia, we further assessed the ability of corrected HSCs to differentiate into myeloid lineage using a conventional CFU assay. Our results show that the frequency of myeloid progeny obtained for uncorrected and corrected HSCs were highly similar, showing that our process does not affect the capacity of HSC to differentiate. Having demonstrated that, we also checked the presence of corrected events within the BFUE, which are the progenitors of red blood cells. We did that by ddPCR analysis, and our results show that more than 50% of the BFUE clones contain at least one allele corrected for HBB, and only 10% displayed the collateral effect potentially associated with beta thalassemia. We then checked whether this efficient genetic correction could be translated into an efficient rescue of functional hemoglobin. To do so, we further differentiate the BFUE colonies into red blood cells and determine the amount of hemoglobin produced by those red blood cells. Our results show that the gene correction enables, number one, to efficiently decrease the proportion of sickle hemoglobin from 90% to 15%, and number two, to enable the production of up to 80% of functional hemoglobin, a value that is way above the limit of therapeutic benefits assessed by clinicians. So we believe that our process is efficient enough to go to clinic, but before doing so, we assessed the ability of corrected HSCs to engraft into a mice model named NSG. Patient HSCs were corrected by our process and injected IV into NSG. After 16 weeks of engraftment, the mouse bone marrow was extracted and analyzed to determine the mouse to human cell chimerism. Our results show that number one, engraftment of corrected human CD45 cells were highly similar from the corrected to the uncorrected cells, indicating that our process did not affect the capacity of corrected cells to engraft. And number two, we had a look at the stability of the gene correction process after 16 weeks of engraftment. And our results showed that the frequency of corrected cells before and after injection were quite similar, showing that the HBB correction was pretty stable over the course of this in vivo experiment, with about 30 to 35% of gene correction in HSCs. Regarding the collateral effects, we were also able to show that it does not enrich during the course of this experiment, showing that there is no proliferative advantage for cells displaying this collateral effect. In conclusion, we've optimized a gene editing process that enables in HSCs to precisely correct the mutation associated to sickle cell anemia. This gene correction is highly specific and efficient. It does not require any enrichment step. It rescues functional and therapeutic level of hemoglobin in red blood cells. We believe that alglobin 01 will provide a significant therapeutic benefits to patients, and it is now poised to move forward for clinical application. Thank you very much.
Hello, my name is Eduardo Cyclone. I'm a team leader here at the Innovation Department in Cellectis. I'm very excited to be here to show some of the early discovery data on the cure of lysosomal storage diseases using HSC genome surgery. As a very brief intro, lysosomal storage diseases, also known as LSDs, are a group of more than 70 rare genetic diseases in which a specific enzyme in the lysosome is not fully functional. As a result, waste product accumulates inside cells all over the body, affecting multiple organs and leading to systemic symptoms. Symptoms that start at a very early age and include very important neurological and developmental disorders. These make LSDs highly debilitating and deadly diseases with very early onsets. And even though LSDs are considered to be rare diseases, as a group, they have a relatively high combined frequency of 1 in 5,000 newborns. Unfortunately, most LSD patients do not have an effective treatment or cure available. For the large majority of them with a severe case, an allogeneic bone marrow transplant represents the only potential cure. But of course, finding a compatible donor is challenging, and patients are still exposed to the high mortality of the procedure and the risk of GvHD. For milder LSD cases, injecting the synthetic enzyme could also partially treat some symptoms. But these infusions are not curative and have to be done during the whole patient's life. In addition to the mentioned disadvantages, neurological symptoms remain mainly untreated, given the inability of the synthetic enzyme to cross the blood-brain barrier. In this scenario, we believe that genome surgery on hematopoietic stem cells have unique features that are especially suited to treat LSDs. Why? Well, cells lacking LSD enzyme are able to take it from its environment if a source is available, for example, after an allogeneic bone marrow transplant. But these unmodified cells secrete very limited levels of these LSD enzymes. With genome surgery, we can engineer HSCs and its progeny to secrete and share LSD enzymes at very high levels. Secondly, after the autologous transplant of edited HSCs, the progeny can distribute systemically, reaching all blood and immune compartments, and therefore making the defective enzyme available for all affected cells and organs. And this is a very powerful feature of HSC genome surgery. By editing hematopoietic stem cells, we are spreading cells throughout the whole body. Our HSC produces millions of cells daily. So by editing HSCs, we're reaching every inch of the body. We're vectorizing engineered HSCs to spread a deficient LSD enzyme throughout the body. Very importantly, the systemic distribution includes the brain, as some cells from the myeloid progeny of HSCs establish what's known as microglia, a small group of cells that reside in the brain and helps neurons function. This, added to the highly increased levels of enzyme that modified cells could secrete, could be a strong combo to treat neurological symptoms. So far, we talked about how LSD are devastating diseases with very limited therapeutic options and highlighted how HSC genome surgery could be especially suited for treating these diseases. But how do we want to do that? One option will be to use a safe harbor approach. This is search for a genomic region, presumably safe to insert an expression cassette for the defective LSD enzyme. In principle, this approach could successfully treat LSDs after disrupting the targeted locus, which could be, for example, CCR5. And it will lead to the expression of the enzyme in all HSC lineages, including also early progenitors and primitive HSCs. We have, however, generated an alternative gene editing approach that offers a number of advantages compared to the safe harbor one. In this approach, we target the intron of a myeloid gene to insert a promoter-less cassette encoding for the defective enzyme and take advantage of the cellular splicing machinery to express both the endogenous myeloid protein and the therapeutic LSD enzyme. By targeting an intron, those alleles that, after TALEN activity, don't adequately integrate the LSD cassette will remain functional to express the endogenous protein despite the potential genetic scar in the intron. But we are not only reducing the risk of collateral damage from our gene editing approach, we're also focusing the expression of the therapeutic protein in the myeloid lineage, and therefore preventing the overexpression in more sensitive cells like early progenitors or primitive HSCs. We believe that this technology could be revolutionary for a number of therapeutic applications that could benefit from this targeted lineage expression. We have named this approach artificial exon, or ARTEX, and as you can see in the table, it includes all the advantages that other gene therapy approaches have to treat LSDs over traditional approaches, like the fact of being curative after a single treatment, being able to express and share LSD enzymes at very high levels, and being potentially able to treat neurological symptoms. But with ARTEX, we're also minimizing the risk of collateral damage by targeting intronic sequences. We're minimizing the risk of mutation by using a cassette that does not include an expression promoter. We're also allowing for a lineage-specific expression of any therapeutic protein. And we are eliminating the potential detrimental effect that overexpression could have in these more sensitive cells, like early progenitors and primitive HSCs. Now let me show you some data using this approach. Like for any gene editing approach, our first task was to find a very efficient and specific TALEN pair targeting the intron of a myeloid gene. And we found one that is highly active, which also have an excellent off-target profile. An off-target profile that was characterized not from in silico exploration, but through an unbiased cellular-based approach to identify over 100 potential off-target sites, each of which was later confirmed to be negative by deep sequencing. With such efficient and safe TALEN pair, we inserted a promoter-less cassette in the intron of a myeloid gene in HSCs. Because the myeloid gene was not expressed in HSCs or early progenitors, we differentiated edited HSCs into the myeloid and non-myeloid lineages in vitro. And as you can see in the graph, when using a safe harbor approach, our gene of interest was expressed in both myeloid and non-myeloid compartments. However, when using ARTEX, we exclusively express our gene of interest in the myeloid lineage at levels compared to the highly optimized safe harbor approach, and with no leakage in the non-myeloid compartment. Very importantly, the progeny of edited HSCs maintain expression of the endogenous myeloid protein. We believe that this lineage expression strategy could open the road for many other therapeutic approaches in the LSD field. We went one step ahead with a specific LSD enzyme and demonstrated in a similar in vitro system that we can successfully edit HSCs with LSD cassettes, and that differentiated myeloid cells can secrete 10 to 15 times more LSD enzymes than modified controls. And it is not shown in the graph, but we have also confirmed the potent activity of this secreted enzyme. Moreover, when we injected edited HSCs into an immunodeficient animal model for engraftment, we observed that cells populated multiple compartments, including mouse blood, spleen, and bone marrow, as you can see in the follow-up plots. But more importantly, we were able to find a small but significant amount of human cells in mouse brains, human cells that share some characteristic human microglial markers, ruling out the potential contamination during the isolation of brain cells. So in summary, our in vitro data shows that the myeloid progeny of engineered HSCs secrete very high levels of LSD enzymes, and that engineered HSCs and its progeny can reach multiple organs, including the brain. I would like to highlight how, using this strategy, we could use different cassettes to produce any defective LSD enzyme, a platform in which a single and safe and well-characterized TALEN could be the anchor for many therapeutic approaches to treat LSDs. With this, I would like to finish the presentation highlighting some key elements we developed. Regarding our approach to treat LSDs, we have shown data that supports how our strategy could represent a single intervention to cure LSDs. This includes systemic distribution of the engineered cells with potential to treat neurological symptoms and the ability to focus the expression of this therapeutic protein in the myeloid lineage. But from a broader point of view, we have generated a therapeutic platform that could be easily adaptable to cure many LSDs by simply swapping the correction cassette. This platform could make the development of therapeutic programs very cost-effective, which is especially important when targeting rare diseases like LSDs. With that, I would like to thank you all for your attention, and I'm sure by now you share our excitement in the field of genome surgery in hematopoietic stem cells.
My name is Tony Katoman. I'm director of the Institute for Transfusion Medicine and Gene Therapy at the Medical Center at the University of Freiburg in Germany. And I'm happy to share with you some data that we have achieved together with our collaboration partner Cellectis in the field of genome editing to treat immunodeficiencies. So in our R&D department, we develop innovative cell and gene therapies for 3 areas of interest. We do this for primary immunodeficiencies, we have a program on HIV, and we also have a CAR T-cell program. And I would really like to stress how important it is to set up these programs from the scratch, from the beginning, together with your clinical partners. So we do this with our clinical partners in the Center for Chronic Immunodeficiencies for primary immunodeficiencies as well as HIV, or with our partners in the Comprehensive Cancer Center for our CAR T-cell program. But I also would like to stress the importance of working together with Cellectis, because with our biotech partner, we are able to bring these therapies to the patients. You all know that conventional gene therapy has been extremely successful in the last 10 years. And this is mainly due because the viral vector systems have evolved and they now allow you to transfer therapeutic genes with high efficiency into your target cells or target organs. Ex vivo, mostly lentiviral vector systems are used, and in vivo, mostly AAV vector systems are used. And this also is reflected in the many successful clinical trials in many different fields. These are gene therapies based on gene transfer in hematopoietic stem cells, like for primary immunodeficiencies or beta thalassemia, but also, of course, in the CAR T-cell field, and also in vivo gene transfer to certain cells of the eye or in the spinal cord have been very successful. And this also transferred into the marketing authorization of now 9 different therapies in the European Union. So if all works so well, why do we need genome editing? And of course, the answer to that is rather simple. Conventional gene therapy works well if you just have to add a therapeutic gene to a cell and this then takes care of the problem. But this is not the case if your problem is, for instance, a gain-of-function mutation. It doesn't work if the level of transgene expression is extremely important. A little bit too much will cause problems, a little bit too little will cause problems. Or, as a third point I would like to mention, when the regulation of transgene expression is extremely important, so meaning you need a certain amount of the protein in a very short window of time in a very specific cell type. And I think this can only be achieved with precision genome surgery or genome editing, if you like, which allows you to correct genes, but which also allows you to knock out genes. Again, this works quite efficiently, and a lot of clinical trials are underway. And I would just like to show this overview here. You see the 3 major platforms which have been translated to the clinic, so zinc finger nucleases, TALENs, as well as CRISPR-Cas nucleases. And you see here the number of clinical studies which are ongoing or already terminated. And you also see an opportunity, a window of opportunity. Primary immunodeficiency is not on the list here, and the HIV trials have not been successful so far. So what do we have to do? If you want to edit the immune system, you usually target the hematopoietic stem cell or
the T cell compartment here further downstream, because all of the immune system actually derives from these hematopoietic stem cells, which you find in the bone marrow of the patients. So I would like to start out with our program on Rag1 deficiency. So Rag1 is a very important gene in the development of B cells and T cells, the two major factors in our immune system. And if patients have mutations in the Rag1 gene, they cannot produce B cells or T cells. And of course, this leads to a severe deficiency of the immune system. This is also the name here, severe combined immunodeficiency. So these patients have to be protected from their environment because they don't have an immune system. And without treatment, they usually die within the first year of life. So why Rag1? Rag1 is, as I mentioned before, only expressed during a very short window in the B and T cell development. It's a DNA cleaving enzyme, so as the nucleases itself. So you only want to have expression in this window of time where it's needed, but you don't want long-term expression of this gene. So, as I mentioned before, you can only achieve this by genome editing and not by conventional gene therapy. So what we aim at here is to really correct the Rag1 gene in patient cells in the hematopoietic stem cell compartment. And of course, the final goal is to restore B and T cell differentiation and so restore the whole immune system in those patients. So just a quick look at the Rag1 gene. It's a very simple gene, just 2 exons here, and the whole coding sequence of the gene is on exon 2. And when you look at the mutations which are found in these patients, you can see they're scattered all over the coding sequence, so there is no hotspot. So our strategy has to be one that can cover all the mutations. So we aimed at integrating a therapeutic cassette upstream of all the mutations, and this is why we target intron 1 with our talents. And this is this nice collaboration with Cellectis. They were able to produce highly efficient talents, as you can see here, which are able to cleave more than 90% of alleles in hematopoietic stem cells. This is what we try to do. So we created a smart donor cassette, which is integrated, as I said, upstream of all these mutations here. And because we integrated into the intron, we have to include a splice acceptor site, which then enables splicing from intron 1 to our transgene cassette here. And I don't want to go into details, but you can see that the choice of a splice acceptor was important, and we found one that mediates very efficient splicing and therefore keeps the transgene expression level high when it is needed. So of course the aim is to integrate this cassette into the correct target site in hematopoietic stem cells. You can see this here. We get about 25 to 30% target integration in the stem cell compartment, and even in the long-term repopulating stem cells, and these are the cells that the patients need to have a long-term effect, we get about 20 to 25% target integration. When we now check whether these stem cells are still able to differentiate into the different lineages, so the different immune cells, we see that there is no significant difference between corrected cells and untreated cells. So I think this looks very nice as well. So just to summarize where we stand in this project is we have highly efficient talents at hand with a very high specificity as well. We have created a smart donor which allows for regulated expression of the Rag1 gene. We see target integration of this smart donor in 20 to 30% of stem cells. We believe that this is now ready for preclinical in vivo evaluations, meaning that these stem cells are now taken to a mouse model to see whether also in vivo these cells can differentiate now after being corrected in B cells and in T cells. A second project I would like to introduce is for patients or for treatment of patients which suffer from a disorder called hyper-IgE syndrome. So the mutations in these patients are found in a gene called Stat3, and Stat3 is a protein, which is important for signal transduction from the surface. You can see here this will be an immune cell, and the immune cell has to let's say, read different signals from the environment and to translate these signals into a response, into a genetic response. So meaning that Stat3 is important for signal transduction from the cell surface to the nucleus, where then different genes are activated in response to this signal. And of course, if Stat3 is missing, the signal transduction does not work anymore. And the consequence for this in that context here is that one of the key elements, a protein called IL-17, is not made anymore. And IL-17 is very important for differentiation of TH, so these T helper cell, to a subclass of T helper cells, which is called Th17 cells. And without these Th17 cells, the patients are not able to fight infections with extracellular pathogens, like fungal infections here, or like bacterial infections in the lung. And on the other hand, the protein is also, or these Th17 cells are also important to regulate the immune system. So we have also overactivity of the immune system as seen here. These are very severe eczema that these patients suffer from as well. So what I mentioned before, the level of Stat3 expression is extremely important. A little bit too much pushes proliferation and increases the risk of cancer. A little bit too little is what our patients have. It's an immune defect. And the second point I would like to mention is that this Stat3 protein comes in two isoforms actually, called alpha and beta. So alpha is an activator of a target gene expression, and beta is a repressor of target gene expression. And again, you need to make sure that the balance between alpha and beta is maintained after genome editing. So what we aim at in this project is to correct the Stat3 gene in the T cell compartment. Again, with the hope and the aim to restore differentiation of these T helper cells into Th17 cells. When we look at the gene again, and we see that the mutations again are scattered almost over the entire gene, but it's not the entire gene. You can see that most of the mutations are downstream of exon 9 here. And this also explains our strategy here. We would like to integrate the transgene cassette upstream of exon 10 using, again, highly efficient talents. And Cellectis, again, was able to produce very efficient talents. You can see here, we target intron 7, intron 8, or intron 9, and all of these talents showed high efficiency in cleaving the target site as well as high specificity. Again, we made a smart donor, which is now integrated into these introns, and you can see here, we get very high efficiency in integrating this donor into the different target sites, up to 80% in intron 7 and about 60 to 70% in the other two sites. So the question is, do they now express Stat3, and do they express the correct ratio of Stat3? And the answer is yes. The transgene is expressed after integration. You can see this here. And with the highest amount of donor, we get about 50% of all the transcripts in the cells are now made from our transgene cassette. And when we look at the ratio between the alpha and the beta form, and you can see this here more clearly, for integrations into intron 7 and intron 9, we can restore the correct balance between alpha and beta. It's about a 1:4 balance, so this looks very promising as well. So what we can conclude here is that we have highly effective talents, we have highly effective integration into the target sites, and we maintain the correct ratio between the alpha and beta form for integrations into intron 7 and intron 9. So the next question is, does this also work in patient cells? So as I said, we have a patient cohort here in Freiburg, and we did this in 3 different T cells now from those patients. You can see the talents work very nicely in those patient cells as well. We get very nice integrations in those patient cells as well. And we also get the correct ratio again between the alpha and the beta form. And just to conclude this part here, I think also in patient cells we can confirm that we get the correct ratio between alpha and beta, and we think that this is ready for clinical development to go as fast as possible to the patients. Let me introduce you to my last project, which is a treatment for HIV patients. You might know that HIV infects two types of immune cells. These are the macrophages here on the left side and the T helper cells here on the right side. And they can do so because these two cell types express a protein which is called CCR5. This is a co-receptor which the virus needs to enter the cell. And of course, if you now want to induce HIV resistance, one approach would be to knock out the gene which encodes for CCR5. And this is what we aim to do in the hematopoietic stem cell compartment, because this ensures that both sides of the immune system do not or are not able to express CCR5, therefore confers the resistance to HIV infection on both sides here of the immune system. It's important to know that, at least in Europe, you don't need CCR5 to live. CCR5 is somewhat redundant in the immune system, and you can live just fine without CCR5. So does it work? Again, we got very nice talents from Cellectis. We show target knockout in about 90% of cells. We can then differentiate these cells again to the different lineages. We show there is no difference between CCR5 knockout cells and normal cells. And when we look now at cell clones, we can actually confirm that we have about 80% of cells where both alleles, so both CCR5 alleles, are knocked out, and in only 5% of cells, none of the CCR5 alleles were touched. I think it's also important to see that when we take these CCR5 knockout cells, we transplant them into mice, and we take them out after a little while, we can show that CCR5 knockout is stable over time. So to conclude this part here is we can show very efficient CCR5 knockout, 90%. The CCR5 knockout is stable over time. It's also safe. We don't see significant off-target activity. And it's also important to keep in mind that the T cells that we generate with the CCR5 knockout are resistant to HIV. So we believe that this whole package shows that this is ready for clinical translation. So let me summarize what I told you. I showed you proof of concept that it's possible to edit the immune system with high efficiency. Our proof of concept studies shows that we can treat primary immunodeficiencies. I showed you one example, Rag1 correction in hematopoietic stem cells or Stat3 correction in T cells. And I showed you a third example as a treatment for chronic immunodeficiency like HIV infection by knocking out CCR5 to induce resistance to the virus. The foundation or the basis for all of this was highly active and highly specific talents. All of them worked really well with about 90% editing efficiencies in primary cells. The second part was to create smart donors that you integrate into the target sites that keep the regulation of the transgene expression. And the third part was to optimize the culturing conditions of these cells. Only this allowed us to achieve these high knock-in frequencies. I mentioned this before, 20 to 30% in hematopoietic stem cells or 40 to 70% in the T cell compartment. All in all, I believe this creates great opportunities to treat patients with an unmet clinical need. Thank you very much.
I hope you enjoyed this last episode dedicated to our next generation of gene therapy products. We are very excited about this new adventure HEAL by Cellectis. To finish, I wanted to share with you our pipeline covering a broad spectrum of genetic disease, including sickle cell anemia, our most advanced program, lysosomal storage disease, and different primary immunodeficiencies. Please join us for the Q&A session for more information. Thank you very much for your time.
Thank you so much for your time for this journey at Cellectis. I hope you have enjoyed the visit. From bench to bed, our innovation cycle is becoming more comprehensive. Our potential is limitless, and we're totally focused on the execution of this tantalizing task. Our target is to become a one-stop shop from the ID to a commercialized therapy. Next time, will tour you for real. Thank you and see you soon.
Hi everyone, and welcome back to episode 5 of the Cellectis Innovation Days. We are so excited to finally be able to talk about HEAL and what it means. I know there's been a lot of anticipation for this episode. Again, my name is Simon Harness. I'm the Chief Investment Officer at Cellectis, and I'll be hosting the live Q&A. And we have a lot of questions already in the queue here. And again, feel free to submit more questions through the question box in the live webcast. So to kick this off today, I think what's most helpful is to summarize a few questions we have gotten that all kind of address the same question. And I will give this one to Julien Valton. Julien, can you describe in a few words on a high level the landscape in sickle cell disease treatments today, coming from AAV approaches to gene editing approaches? And in gene editing, what are the different current approaches that we see in the clinic, and how Cellectis differentiates itself with the correction to the natural DNA of our program?
Thank you very much. I'm Julien Valton, the Vice President, Gene Therapy. So, very interesting question. I think, first of all, I'd like to start by congratulating our colleagues at Bluebird Bio and Vertex and CRISPR Therapeutics to set the stage in the gene therapy of sickle cell anemia. I think they have achieved tremendous, very good results. And to answer the first question, I have to say that the main difference between all the different approaches by the colleagues is that they are not correcting the mutation associated to sickle cell anemia. We do correct the sickle cell anemia mutation. So to get much more granularity into that, you have on one side Bluebird Bio that vectorized the correct version of HBB using lentiviral particles, and you have on the other side CRISPR Therapeutics and Vertex that are upregulating or promoting the upregulation of HbF, which is a surrogate version of the adult hemoglobin that is missing in the sickle cell anemia. We do not want to go with this. We want to go for the GRAIL, and we want to correct the function and recover the actual function of hemoglobin. So that's, I think, the main difference between the competitors, our colleagues, and us. One thing I can...
Thank you. Yeah, go ahead.
One thing I can add up, one thing I can add up is that because we are correcting the HBB locus, the correction HBB is under normal regulatory conditions. So it means that the HBB will be expressed at a physiological level, which is not the case for RLV approaches pursued by Bluebird Bio and not the other approaches. So that was the other point.
Thanks, Julien. And can you also go into more details about our advantages in our sickle cell disease approach compared to strategies that target BCL11A? I know you already talked a little bit about the competitive landscape, but just more in precise manner on this.
Yeah, so I think like BCL11A, so if you inactivate BCL11A, you upregulate the HbF. And again, HbF is just a surrogate version of the HB adult that is missing in this sickle cell anemia patient. So it's on one side we have our approach where we correct the actual HBB and recover the HB adult hemoglobin, and on the other side you just upregulate a surrogate version. So because those two proteins may not have the same function, like exactly the same function, we think that we are we go for the best-in-class approach. Also, the other difference is that all those different approaches, the outcome of all those different approaches, you also have HbS still lying in the cells of patients. We do not want that, and that's why we went for the gene correction, so that we do not have any more HBS lying in the cells of patients.
Simon, if I may add something on this very good question, because of course it raises a lot of questioning around it. At the end, when you speak about gene editing, everyone would understand it, or the patient would understand it by going inside the cell and fixing the mutation. This is what we do. We get there, we go on the physiological HBB gene that is altered with a spelling mistake in there, and we fix it back. Everyone understands gene editing is this. When you go and see a patient, you tell the patient, you have three options here. First option, I'm going to insert another gene somewhere randomly in your genome. Or the second option you have, I'm going to destroy a gene, I'm going to reactivate another gene that's not supposed to be activated, but the mutated gene will stay there, etc. Or you say, just going there, and I'm going to restore the mutated gene. This is what we do. And that's what everyone understands what gene editing is. And if you want to project yourself over this decade, this is what gene editing should be.
Thank you so much, Andre. There's a lot more to cover, so I'm going a little bit into the technicalities of the gene editing piece. So questions from Yigal at Citi, from Jack at Baird, and from Gina at Barclays. First of all, are we using TALEN, and why are we using TALEN in this program? And the second question is, which component will be packaged in AAV6? And the follow-up question is a 50% correction sufficient for clinical benefit, and what is the minimal bar? I'll give this question here to Patrick and Eduardo.
Hi, thanks for the question. My name is Patrick Hong. I'm a team leader in the innovation department. The first question, although we have years of experience in the other technologies such as CRISPR, we are using for the genome surgery, we are using the TALEN technology. And depending on the strategy, whether it's a gene correction, insertion, or disruption, the TALEN technology that we are using is very comparable to the CRISPR system, and in some cases, the efficiency is actually better. And I can actually refer to the second question to my colleague.
Hello, this is Eduardo Cyclone. I'm a team leader here at Innovation. So yeah, just following up on the second question for the AAV. We are delivering currently like the donor template by the AAV using the serotype 6, which is, as you probably know, like very highly effective into delivering the cargo into hematopoietic stem cells. Not only that, it's really good compared with like other delivery methods to support homologous recombination or like correction in general for gene editing. So that's why we chose to use that delivery vehicle.
Thanks, Eduardo. I actually just have a follow-up question from William Blair. Are you able to filter out cells that have collateral HBB knockout instead of gene correction so they will not be part of the final product?
I can take that.
Maybe Paris.
So, yeah, Julien here. We are not able to sort out cells that contain a collateral effect, and that's why we optimize the process to prevent this event to occur as much as we can. And so today we have like less than 10%, 10 to 20% of collateral effect. And 10% of biallelic collateral effect, which is way lower than what it was expected before optimization.
Thanks, Julien. And a follow-up question from Gina at Barclays, and this is for Eduardo. For LSD, I assume you use integration to insert transgene to specific sequence. Will that be done ex vivo with electroporation together with donor sequence packaged in AAV6? What is the cell survival rate? How consistent is the insertion rate across different programs? Which are the high interest spots for insertion? Albumin intron 2 was a top choice for other companies. Any thoughts on this and beyond?
So thank you for your question, Gina. So yes, we are currently using the same ex vivo platform for editing, also for the LSD therapies, which consists, as we mentioned before, into like delivering the TALEN by electroporation and the donor template by AAV. We're currently reaching the same levels, high levels of gene editing rates that we reach for the HBV program, reaching this 60-70% editing rate in this myeloid gene. And following out on the cell survival and viability in general is also pretty high, like 80-90%. So we're having a pretty safe and efficient platform also for these diseases. And then on the second part of the question was more about like the gene editing approach that we're using. So remember, like in this case, we're not changing just one base pair. Like for the HBV program, we're inserting a large cassette. That's in the intron part of a gene. And you are mentioning like albumin as a potential target for that, like other companies are using. These companies are probably using albumin because they focus into treating hepatocytes or changing hepatocytes, which express a lot of albumin. HSCs do not express a lot of albumin. So that is the reason we're focusing on other genes on HSCs that are expressed more in the progeny of HSCs. We want to prevent the expression of these genes in early progenitors or like in pluripotent cells like stem cells. So again, we're focusing our editing into a myeloid gene, in the intron of a myeloid gene, as opposed to albumin. And it's, as you've seen, some of the data is being pretty efficient.
Thank you so much, Eduardo. I'm going to pivot a little bit into the clinical questions for our Chief Medical Officer, Dr. Carrie Brownstein. Starting with a question from Jack at Baird. How are you thinking about the preconditioning regimen needed prior to administration of your sickle cell therapy? I'm aware that there are a number of various regimens being tested in this space. Are you considering options to minimize the use of chemo preconditioning?
Yeah, so this is Carrie here. Thanks for the question. I think, there are, as you said, there's lots of ways of looking at how to precondition. Given the fact that these are autologous products, the preconditioning would not be as severe or myeloablative as you would see in a patient who is getting an allogeneic stem cell. So typically the patients who would be sent to this program for a clinical trial are going to be the most severe patients who normally would be eligible for a hematopoietic stem cell transplant, but because of whether they don't have a donor or other reasons, wouldn't be getting one. So we would not be expecting the preconditioning to be anywhere near what they would have been required for an allogeneic stem cell transplant. And we're currently working with our collaborators in the clinical side to come up with what we think is going to be the best and most efficient regimen for the program.
Thank you, Carrie. And also a question from Ingrid at Kempen. How do you think that the clinical development will look like for LSDs? Could you use several indications in one trial potentially, or build up a bridge or pivotal trials on top of the same phase 1 or 2?
Yeah, so I love this question because I am a big proponent of the master trial programs. I think that generally speaking, the particularly large pharma has not done a very good job of finding ways to be efficient and cost-effective in the development and of products for rare diseases. So I think it's really important that we, a priori, for many of these programs, and not just for the LSDs, but for when we get into many of these programs for rare diseases, it's critical that we find solutions like master trials where we can look at not only slightly different versions of the same product in one patient population, but also look at slightly different patient populations with different products so we can do things quickly, efficiently, and in a nimble manner so we can bring things to patients as quickly as they need.
Thank you, Carrie. Sure. And just as a follow-up, what's your current program to get this into the clinic? What's our process? And maybe some rough timelines. I know this is maybe a little early, but just like what process are we in right now?
Right. So for these programs, we're still, and particularly, we're talking with our potential clinical collaborators so we can find ways to move forward as quickly as possible. We don't have a specific time frame right this minute.
Thank you, Carrie. And yeah, we'll share more on this as we obviously progress through manufacturing and through our discussions with our upcoming clinical center. So I'll make sure to communicate those timelines as soon as we have them. I'm going to pivot really quick into a few questions for Dr. Tony Kutterman. Thank you again so much for this wonderful presentation today and for joining us. So we have a few very specific questions here. I'm going to start with a question on Stat3. First of all, for Stat3 and Rag1, what is the size of the market do you expect? And a follow-up question unrelated to this, but on Stat3, what can we share about the mouse models, and is there an efficient mouse model for Stat3?
Yeah, thank you very much for this question. I think I will start with the second question. I think there are a couple of Stat3 knockout mouse models, but none of them are suited to really test the Stat3 gene-edited human T cells in vivo. So what we have planned to do in this context is to apply for clinical trial authorization without the mouse data. In terms of the market size, I think Stat3 deficiency as well as Rag1 deficiency, these are very rare disorders. So I think about 1 in 100,000 to 1 in 1 million births are affected. So you can do the math. I think this is really rare diseases. But what I have to add to this is these are also prototypical disorders of the immune system. So whatever we develop, whatever the technical advances that we make for those disorders, they can be expanded very easily to other disorders of the immune system.
Thank you so much, Tony. I'm going to pivot back here into these programs from a manufacturing and IP standpoint. So thank you, Ingrid from Kempen, for this question. We heard a lot about your manufacturing and underlying technologies in the past week. Now that you're moving from an allogeneic strategy to also have autologous candidates, what does that change in terms of our manufacturing capabilities and underlying technologies? How much do we need to adapt, and how much can we leverage? And this is a good question for Paris.
Yes. So thank you for this question. This is Leo Berthier, SVP Tech Ops here in Paris. So basically, we set up our manufacturing capability both in US and in France with the idea to be very flexible. So we can do both allogenic and autologous projects in our capabilities. So it's really, it has been designed with this purpose, and that's the way we will handle it. Thank you.
Thank you very much, Leo. And a question from Gina Wang at Barclays. What kind of IP do we have on this approach? If other gene editing companies want to use similar approach of cutting an insertion from AAV donor, how do you protect your technology or differentiate your technology? Good question for Paris. Andre, I don't know if you want to answer.
Well, we have series of very strong patents in this field, especially like umbrella gene editing patent covering this. Of course, there is always a series of different patents covering different type of technologies. Most of the time, these patents are still patent applications, and you have to see what comes out from it. So far, I think that we have to be very cautious, and IP is always needed in this space, but it's also restricted most of the time to the technologies that are used. For example, we own series of patents on TALEN technology, like we've shown it on CAR-T, but we also own same type of patent, but for CRISPR technologies, also on CAR-T, etc. So the patents tend to be down to the USPTO or the EPO, like European Patent Office, more and more restricted to the technologies that are used. And one of the good things we have on our side is that we're very technology agnostic, and we've been filing patents on series of different type of gene editing technologies, and that's helpful also. On the AAV side, there is also a series of patents and other type of patents that have been used. The AAV is an old technology and has been used for years and years, and a lot of patents patents have been off patent today, and still it's a moving part, so we'll see at a time where we'll commercialize one of these products.
Thank you very much, Andre. We're going to pivot back a little bit into the gene editing process, and I have a few questions for Julien. First question from Gina Wang at Barclays. For this new technology, is it a one-step process, meaning that talent cutting an AV6 donor in the same step? What other editing have you seen other than proper insertion?
Thank you, Simon, for the question. So it's a two-step process where we electroporate the cells and then add the virus onto the electroporated cells. So that was the first question, and the second question is regarding the specificity of the correction matrix insertion. So far, we haven't seen any upside insertion of this AAV6 matrix. And I think that was the question, right? Yeah. So
Thank you. Thank you very much. And just as a follow-up, Nick Abbott from Wells Fargo's question, are HSCs isolated before genome surgery, or is there a strategy to target the HSC in a bulk population?
So the HSCs are isolated through apheresis, then we purify them, CD34 positive purification, and then we edit them. So we edit a bulk population somehow of CD34 cells, which is the common approach used by other colleagues. And it's overall a 4 days process where we collect the cells, expand them, electroporate them, and then let them grow for an additional 2 days before potentially injecting them into the patient.
Thank you very much, Leon. And also a question from William Blair. Are you able to sort or filter out cells that had collateral HBB knockout instead of gene correction, so they will not be part of the final product?
So I think I answered this question already. So no, we are not able to sort out the cells. And that's why we de novo implemented an optimization process so that we decrease as much as we can the cells containing this collateral effect by playing on the editing process. So no, we are not able to sort out the cells for the gene correction process. But for example, for the LSD project, we could potentially add a cassette that is expressed at the surface of the cells to fish out the cells that contain the corrected event. So it's different type of approach. We can adapt, actually.
Thanks, Julien. I just wanted to make extra sure to ask the same question twice. I'm sorry about that. I have another question here for Eduardo from Wangzi Li at Ladenburg. For myeloid secretion of enzymes to treat LSD, is there a risk of overproduction of the enzymes from the myeloid cells persistently?
So thanks for the question. I think I can answer that in giving you 2 pieces of information. First, LSD enzymes are expressed in all cells of the body. So not only everywhere, from stem cells to muscle cells, everywhere. They do need to be integrated in the lysosomes to process products. So from that point of view, it shouldn't be toxic. And then the other piece of information that we could get is there are other therapeutic approaches that are currently using lentiviral vectors to overexpress these enzymes. And so far, there are no adverse effects observed in these trials. So I guess we don't expect to see the same in our gene-edited approach for LSDs.
Thank you, Eduardo. I have another question from Nick Abbott at Wells Fargo for Patrick. I thought I saw there was a 4-fold increase in HSC editing efficiency versus standard. If correct, how is that achieved?
Hi, Nick. Thanks for the question. Actually, I have to make a correction for you that the presentation that we showed you today is not a correction, a 4-fold increase in the LT-HSC correction. It's actually expansion of LT-HSC is increased by 4-fold. and we can actually achieve this in the either in the R&D and GMP process, with our GMP grade materials.
Thank you, Patrick. This concludes today's question. Oh wait, I have one more question. Sorry, from Gina. Following up question on editing. Beyond correct insertion, did you see AAV integration to the cells? What other modification have you seen and what percentages? Maybe Patrick, that's a question for you. Beyond correct insertion, did you see AAV integration to the cells? What other modification have you seen and at what percentages?
The data that I showed you today was the gene correction by AAV, introducing a repair matrix by AAV, but also the gene insertion rate was achieved over 70%, and also the disruption rate without AAV was much higher. So we have depending on the strategy, we can actually achieve at the minimum of 70 to 75%, and up to over 90%.
Thanks, Patrick. And I know we're already over time, but we really appreciate all your questions, and feel free to, again, send them to us directly as a follow-up. We'll be talking much more about this program now going forward, and we're excited again to share this today for the very first time, which we think is the first real curative potential approach in this disease. So thank you very much for the questions, and again, we'd be happy to jump on the phone with you guys directly to answer these questions. Talk to you soon, and thank you.